Hello, students. Today we'll explore the biochemistry of glutamic acid production, a process that makes one of the most important and widely produced amino acid in industrial biotechnology. Glutamic acid is a non-essential amino acid. It plays a central role in protein synthesis and in nitrogen metabolism in the human body. The sodium salt of glutamic acid is called monosodium glutamate, or MSG, is well-known flavor enhancer that gives food its distinctive umami taste. Therefore, MSG, often described as the fifth basic taste, along with sweet, sour, salty, and bitter. Now let's take a closer look at the process, how glutamic acid is produced using microbes and the biochemistry behind it. The process begins with choosing an appropriate carbon source, such as glucose, molasses, or other sugars, which serve as growth substrate. When bacteria are grown on glucose, they generate energy and metabolic intermediates. Through a series of enzymatic steps, glucose is broken down into two molecules of pyruvate. Therefore, one glucose molecule breaks down into two pyruvate molecules, producing two ATP and two NADH molecules. In next step, pyruvate enters the tricarboxylic acid, or TCA cycle. In TCA cycle, pyruvate is first converted to acetyl-CoA. In this reaction, pyruvate combines with CoA and NAD+ to form acetyl-CoA, carbon dioxide, and NADH. In second step, acetyl-CoA combines with oxaloacetate to form citrate. Citrate is then converted into alpha-ketoglutarate, producing NADH and releasing carbon dioxide. The critical step is the amination of alpha-ketoglutarate. The enzyme here is glutamate dehydrogenase, or GDH. The substrates are alpha-ketoglutarate, ammonium ion, and NADPH. The products are glutamate, NADP+, and water. Here, NADPH acts as the reducing power, which is supplied by the pentose phosphate pathway. So we see that carbohydrate metabolism, including glycolysis and the TCA cycle, and nitrogen metabolism converge at this point. So, under normal conditions, bacteria do not actually release glutamate. But to induce secretion into the broth, there are a few strategies. Limiting biotin, for example, weakens the cell membrane, which allows glutamate to leak out. Also, adding mild detergents or surface active agents like tween-80 or penicillin can increase the membrane's permeability. And finally, there are mutant strains of Corynebacterium glutamicum that are specifically designed for continuous glutamate secretion. Besides glutamate dehydrogenase, glutamate can also be formed through the glutamine synthetase glutamate synthase pathway. So, here's how it works. Glutamine synthetase, or GS, catalyzes the reaction where glutamate and ammonium ion combine to form glutamine, and this step actually requires ATP. Then, glutamate synthase takes over. Glutamine reacts with alpha-ketoglutarate and NADPH to yield two molecules of glutamate. This pathway, by the way, is especially important when ammonium concentrations are low. Let's sum up the pathway real quick. Starting from the carbon source, usually glucose, it goes through glycolysis to form pyruvate, which then enters the TCA cycle to produce alpha-ketoglutarate. For nitrogen assimilation, ammonium ions come into play. The key reaction is when alpha-ketoglutarate, ammonium ion, and NADPH combine to form glutamic acid. The main enzymes involved are glutamate dehydrogenase, glutamine synthetase, and glutamate synthase. Regulation of this process depends on a few factors like membrane permeability, biotin concentration, and even strain mutations. So, in conclusion, the production of glutamic acid is really a beautiful example of microbial biochemistry. It brings together carbohydrate metabolism, nitrogen assimilation, redox balance, and even membrane engineering to create a compound that honestly touches so many aspects of our food, health, and environment. Thanks for watching. If you enjoyed this video, please like and subscribe for more.